Hello everyone, I am Ajesh P. Thomas from the Department of Chemistry, University of Kerala. The title of this lecture is Porphyrin, the Pigment of Life. The porphyrin, maybe some of you might have heard the term porphyrin, or maybe if I show the structure, you may identify, okay, this is porphyrin, or some of you may be expert in porphyrin. So whatever it is, from this title, it's clear that it's a pigment and is something related to life. So today I will be going to give a brief outline of porphyrin. I will not be going to the details of much details of porphyrin chemistry, but what I can do is I can give a outline of porphyrins and its significance in biology, in living systems, and its advancement. Looking at this slide, I just kept some of the figures that are relevant to our biological systems. Say a juice of beetroot and the flamingos and vegetables in say yellow colors and the iris and some other vegetables. So all what you can see is different colors of vegetables and animals and related to our living systems. And of course, these different systems or all these different colors are coming from different organic compounds. For example, in the case of beetroot, it is betalains, and for say flamingos, and in the case of the carrot and other vegetables, it's coming from different beta carotene derivatives. And in the case of iris, the color of iris is because of melanin and the different composition of melanins, different type of say different analogues of melanin, I would say. And again another set of vegetables including grapes and strawberries coming from anthocyanidin family organic molecules. So this indicates that behind the coloring of different objects, some organic molecules, some organic dyes are involved. But here in this lecture, we are trying to focus on in another dye, not these molecules. We are interested in another dye. I'll come to that. That is porphyrin. So why as my title indicates, the porphyrin, the pigment of life. So it is known as porphyrin is known as the pigment of life. So the question is why? So when you look at this slide, what you can see is a red blood cell and a green leaf and a vitamin B12, say cobalamin. So you might have known the importance of red blood cell and the hemoglobin. The red blood cell contains hemoglobin and its role in the oxygen transport and oxygen storage, oxygen transport in the form of hemoglobin and oxygen storage in the case of say myoglobin. And coming to the green color of the leaf, you know, it is because of the chlorophyll molecule, and it is very important in the photosynthetic process. It is the key in the maintaining of the biological or the living systems where the carbon dioxide and water combine together to form say carbohydrate. And another picture that I given is about cobalamin, vitamin B12. It is important in the case of different metabolic process and its function in the nervous system, and its deficiency will lead to different health issues like say anemia and nervous problems, nerve problems. All right. So the point is the color or the basic molecule in all these red blood cell and green leaf and vitamin B12 is a ring molecule which is known as porphyrin. So this is the structure of porphyrin given as shown here. Is a tetrapyrrolic system. I will come to that, the details of the structure. I will come into the next slide. So as you can see, is a ring. Simply what you can understand is it is a ring. Is a ring molecule. Is a ring shaped molecule. And this is what you can see is the hemoglobin with the porphyrin molecule with a metal ion at the center, and in the case of chlorophyll, again a porphyrin like structure with the magnesium at the center, and in the case of the vitamin B12 cobalamin, where you can see a cobalt at the center of a porphyrin ring. And porphyrin can porphyrin present in different biological system in different forms, not only in this particular form, particular tetrapyrrolic system, it's a highly conjugated system, but also the given below is another forms of porphyrin known as chlorine, bacteriochlorine and isobacteriochlorine. So the chlorine structure you can found in the case of say chlorophyll. Chlorophyll you can found that it is not exactly the porphyrin, it's a derivative of porphyrin, we call it chlorine. So these are the structures, porphyrin based structures that is present in the different living system and that contribute significantly to different biological process to maintain the life. The story of porphyrin, before I go into the further details, the story of porphyrin starts in the prehistoric time. Because the name, the porphyrin, the name is coming from the term, the Greek term porphyrus. They used that term to represent the purple color. Porphyrin having a very intense, normally the tetrapyrrolic porphyrin I am talking about, a intense purple color. So the name coming from this Greek word porphyrus. But before they identify this is a porphyrin, the purple color is something related to the royal life at that time. I mean the priest and the kings, all these people used their clothes and based on the purple color. And they extracted purple color from different biological system like molluscs and using some plants to extract this purple color. And as I mentioned, this color having a very significant role or having a royal role in their lifestyle. So that's why the name coming from. So porphyrin is look like a purple colored compound, so that the name is coming from the term porphyrus. Okay, so already I have discussed that, I mentioned that yes, porphyrin and its porphyrin related structures having role in biological system. So the question is how this porphyrin molecule is synthesized in a living system, say in animal system or in a plant system. So the key molecule to make the porphyrin molecule, porphyrin biosynthesis or heme biosynthesis is a delta aminolevinic acid or 5-aminolevinic acid. So this is a common ingredient in the case of plant system and animal system, even if the starting material differ slightly in the case of both plants and animals. This is given for the animal system where you start from glycine and succinyl coenzyme A, and they synthesize the delta aminolevinic acid, and which undergoes a series of synthetic modifications involving different enzymes to make finally, see, I am not going to do all these details, as you can see here. Finally it ends up with a protoporphyrin 9, which is then converted to protoporphinogen 9, which is converted to protoporphyrin 9, which is after metallation using the help of the enzyme, the ferrochelatase, to form say heme B, which is again, as you know, is contributing to the formation production of hemoglobin. The heme synthesis is mainly happening in the bone marrow and in liver. When you say bone marrow, it is mainly related to the red blood cell erythrocytes, and the synthesis happening started in mitochondria of the cell, then it slowly moved to the cytoplasm. So both the mitochondria and the cytoplasm is involved in the synthesis of heme. So this is the case of heme, but same thing will happens in the case of chlorophyll and vitamin B12. Only thing the final, like instead of ferrochelatase to methylating with iron, in the case of chlorophyll an enzyme will work to methylating it with say magnesium. Instead of iron, magnesium will come in the case of chlorophyll. So that is the difference, but the other steps will remain same. So this is a key process happening inside the living system to synthesize the porphyrin derivatives to do their functions. Yeah, this is what I mentioned. This is again a compact form of how a protoporphyrin is formed, starting from aminolevinic acid, undergoing different steps, and finally it can be converted, or it can be, different derivatives can be generated from this, including say chlorophyll, hemoglobin, vitamin B12, and another coenzyme F40, which is related to the bacterial function, which is having a nickel at the nickel at the porphyrin center to do their function. As I mentioned, in a biological system, it's not only about the structure I shown before, like the tetrapyrrolic system. Some other structures also involved, like chlorine and phobine and bacteriochlorine and say bilane. It's a non-cyclic derivative of the tetrapyrrolic compound. And this is a different color. I mean, as I mentioned, the porphyrin is highly colored compound. Even it's generally accepted that it's a purple colored compound. Different derivatives can have different colors. Once I say different derivative, I will come to that, what is the meaning of different derivatives. But here you can see that all are tetrapyrrolic systems, but they are slightly different in their conjugation and substitution, and they can all are related to different biological process, and these are different colors associated with these molecules. You can see the color changing from the purple or red to say yellow in a unconjugated or non-cyclic bilirubin structure. All right, so now we will go to a little bit more details of the structure of porphyrin. So I talked about, okay, I already mentioned that it's a tetrapyrrolic system. As you can see 4 pyrroles. So the fundamental unit in porphyrin is pyrrole. So we can see 4 pyrroles conjugated. You can see a nice conjugation. That's why the pyrrole is a colorless molecule. But once it make a porphyrin using when 4 pyrrole unit combines together, it will become a very colorful molecule. That is because of the highly conjugated system that's in fact contributing to the color of this molecule. Okay, and it can say in two isomers, and this is what I will show, the 2 isomers of this porphyrin, and the red color indicates here is how the electronic conjugation is happened in this tetrapyrrolic ring system. I want to make specifically to show how we name this different position in this ring system. Here these green colored carbons are known as the mesocarbons or meso position of this porphyrin. This is very important when I discuss in the next slides. And these pyrrolic carbons available here, which is I mentioned in the star, that is known as the beta carbons. So when you look at the structure, what you can find is in a simple tetrapyrrolic system porphyrin, we will have 4 meso positions and 8 beta positions. And its importance is that we can make different substitutions at this point to change its physical and chemical properties. So I will come to that. I can give a few examples or how can we change this different substitution in the beta and the meso position. And when we think about the aromaticity of porphyrin, the question is whether porphyrin is an aromatic molecule. The answer is yes. Normally it is yes, but there are some porphyrin derivative which is not aromatic, but we are talking about this tetrapyrrolic system. So when we look at this structure, you can find that we can find 22 pi electrons, 11 bonds are in say conjugation. But in fact, we need only 18 electron in the conjugation. I mean, the 2 bonds can be spared. This is what I mentioned from this red colored say porphyrin ring. So here, as mentioned here, these red colored electrons, I mean the red coloredly marked double bonds are participating in the conjugation to make it say 18 pi electrons are involved in the conjugation of the porphyrin to satisfy the Huckel rule to make it an aromatic compound. And another important point I have to mention is that these protons, these lone pair electron in this 2 nitrogen can be easily protonated with some weak acid like say trifluoroacetic acid, so that we can say it is a slightly basic character. The porphyrin is slightly basic, and because of that, this ring, I mean the core of this porphyrin ring can be utilized for different kind of metallation. All different kind of metals can be occupied inside this core of the porphyrin, and once it's metallated, the property of the porphyrin will entirely change, which can, an example is the one that we discussed earlier, like hemoglobin and the chlorophyll. Both are having similar structure, not exactly same structure, similar structure, but in one molecule, one compound we is having iron at the center, and another case we have magnesium at the center. So but the point is, the core of the porphyrin can be easily modified or easily can be incorporated with different metal ions. A range of metal ions can be incorporated inside this metal center, including zinc, iron, cobalt, and nickel, etc. And that will have, all this will give different properties and different properties. All right. So yeah, here another thing I have to mention is the tautomeric forms of porphyrin. The nitrogen, see, as you can see, is the proton is placed in a trans position in this porphyrin ring instead of the cis position, and is given that the trans position is more stable than the cis position. That's why normally the porphyrin will adopt this kind of structure. I will show you a synthetic protocol from the synthesis of a tetrapyrrolic porphyrin, a simple porphyrin. So before I discuss about the synthesis, I would like to say about a historical background of the synthesis. So as I mentioned, this color, this purple color is very interesting to those people, and is considered as a royal thing, and they extracted this purple colored compound from different living substances. The porphyrin was first isolated from the blood. The liquid blood, when added with some acid, they got some precipitate, and initially they thought that the red color of this blood is because of because of the iron element, but later only it's found that it is due to some aromatic molecule. Initially many people didn't agree that an aromatic molecule, a big aromatic molecule, cyclic molecule can be stable, but later people agreed that okay, the red color of the blood is due to this porphyrin molecule. After that, many synthetic attempts were done to make this molecule in the laboratory, and the pioneer in this synthetic chemistry of porphyrin is Fischer, and he is known as the father of modern porphyrin chemistry. He has done a lot of synthetic protocols, synthetic procedures to make the porphyrin in the laboratory. But this procedure I given here is a simple procedure. There are, as I mentioned, the different. In the case of Fischer's synthesis, he used only pyrrole and acid. High temperature, he is able to make. He will be able to make the porphyrin ring. But the yield was very less. But in the modern synthesis, it has been advanced very much. And here in this synthesis, what I showed is, what we need is an aldehyde, any aldehyde, and in the presence of pyrrole with some acid and some oxidizing agent. Normally the oxidizing agent is DDQ, the structure is given here. So when we treat with this molecule, and after oxidation with the DDQ, we will be ending up with a ring structure, porphyrin ring structure. And as I mentioned, depending upon what aldehyde that you are using, you can modify the meso positions. So the modification in meso position is found to be one of the easiest synthetic protocol that can be adopted to make different type of porphyrins and to easily modify their property. And as I mentioned earlier, also we can modify the beta position, but which is not that easy compared to the meso-substituted porphyrins. So now coming to the photophysical properties of porphyrin, this is given the absorption and emission spectrum of porphyrin. So the question is, what is the reason for the purple color of the porphyrin? So this is due to its very good absorption in the blue-green region of this porphyrin. So as you can see is this band, the very intense band in the case of around 400 nanometer, in the case of tetraphenyl porphyrin it will come around 420 nanometer, and is having very intense absorption in this region, and this band is known as a Soret band. The name of Soret is a name of a scientist, and after that we can see 4 Q bands in the region ranging from say 525 to 650 nanometer. So this is a characteristic absorption peak of a porphyrin, a tetrapyrrolic porphyrin. So when you have other derivative, of course there will be a change in the absorption spectrum, but this is an ideal absorption spectrum of a porphyrin where we can see an intense Soret band and 4 Q bands. And this is the emission spectrum of the porphyrin, where there is emissive normally in the 650 region, and is having very good emission in the very dilute condition. At higher concentration, its emission will be decreased because of other reasons. I am not going to those details, but normally porphyrins are highly emissive, and the emission property of the porphyrin can be used in different applications. I can give a just outline of the different kind of applications where the porphyrin emission properties has been used. In initial slides, I talked about different derivatives of porphyrins, or I use those kind of terms. So what is the meaning of different derivatives? So these are different porphyrin structures or different say porphyrin analogues. So this is the middle one is the tetrapyrrolic porphyrin, and instead of porphyrol, we can make synthetically in the laboratory, we can make a porphyrin ring with 5 pyrrole units. So that we can call as expanded porphyrin as shown here. And in some cases, instead of the pyrrole, we can use other molecule like thiophene or furan. So the core of the porphyrin is modified, and that kind of porphyrins are known as the core modified porphyrin. And another modification is inversion or unconfused porphyrin, where the pyrrole ring, one of the pyrrole ring is flipped. See, when you look at the porphyrin structure, what you will be able to see is all the 4 pyrrole rings are connected by an alpha-alpha dash linkage, and in the case of a inverted structure or unconfused porphyrin structure, one of the pyrrole ring is flipped to make an alpha-beta dash linkage. So it's not only one pyrrole ring, different rings also can be flipped, that makes more confusions. So I am not going to those details. And another modification possible in the structure of porphyrin is a contracted porphyrins. In contracted porphyrin, we can see we can have different type of contracted porphyrins. But here what I have shown is a porphyrin structure with 4 pyrrolic unit, but one of the meso position is missed. Instead of 4 mesocarbons, here you have only 3 mesocarbons. So that means it's contracted a bit, and it's known as a contracted porphyrin. Of course, as I mentioned, the beta position also modified to make peripherally modified porphyrin structures. So the point is that a lot of things can be done in the case of porphyrin structure. We can do different modifications, and most important part is all these modifications will contribute or will give different properties that can be used for different applications. So and another important thing is it is easy to modify. We can make a protocol, we can make a plan to make a different substitute at the meso position, and we can make a plan to make a porphyrin with a expanded porphyrin with 5 rings, 6 rings, and we can make a contracted porphyrin with say 3 pyrrole unit. So there are different modifications that are possible. Here I want to show some of the examples of expanded porphyrin. I mentioned that yes, porphyrin can be modified. Instead of tetrapyrrolic porphyrin system, we can have a porphyrin with 5 pyrrole rings. So this is an example for that. A pentaphyrin we call it a pentaphyrin, and say hexaphyrin 6 pyrrole unit, and see 26 electrons in conjugation to make it 26 electrons are involved in the conjugation, and heptaphyrin with 7 unit with 32 electrons, octaphyrin with 8 pyrrole rings, nonaphyrin, decaphyrin, and bigger than that. So researchers are involved in modifying the porphyrin structures and to extract different properties out of that. So this is examples. In this example, I want to show that different kind of expanded porphyrin derivatives that is reported in the literature. Can give an example of contracted porphyrin, a tribenzo subporphyrin, contracted porphyrin or subporphyrin. Normally, instead of 18 pi electron system in normal porphyrin, in the case of contracted porphyrin, it is say 14 pi electron systems. So the methyl array substituted subporphyrins, and pyriporphyrin, instead of pyrrole, we have a 1 unit of pyridine in the subporphyrin structure, as a tribenzo triphyrin. So different contracted porphyrins also possible, and that will give different properties. So these are just what I have given is just an outline of what different synthetic modifications possible. When you look at the SciFinder, or in case of Web of Science, when you type porphyrin, just keyword porphyrin, you will be able to see thousands of references regarding porphyrin. and most of them mainly focusing on the synthetic modification, as I mentioned. So researchers are broadly involved in the synthetic modifications of porphyrins to extract different properties. But at the same time, the properties of the synthetic porphyrins also widely explored. When I say the properties or the applications, this slide shows the different applications in which a porphyrin molecule is involved. Some of them are photodynamic therapy, photothermal therapy, fluorescence imaging, and PET and MR imaging. So in the medical applications, this is I give only in the case of medical applications. But the applications of porphyrin is not only restricted in medical applications, it is having applications in of sensing. Porphyrins can be used to sense different metal ions, different anions. So it's application in sensing applications. It is having application in solar cells, dye-sensitized solar cell. We are using different type of porphyrin molecules, and porphyrin based nanoparticles having another different kind of application. So the applications are very broad. Here I just kept some of the applications in the biochemistry or medicinal chemistry, and in one of this point in photodynamic therapy, where the porphyrin molecules are widely used, and in one of my next talk I will be talking about photodynamic therapy. So what I want to make is, this molecule can be easily modified, and each modification can be used for different applications in our life. And as apart from the molecule as such, there are different this porphyrin molecule can be integrated with different polymers, and that will give different other applications, as I mentioned, in the case of again biomedicine, in solar devices, in adsorption, different chemical adsorption compounds, and in catalytic properties, etc. So these are the examples where a porphyrin molecule or a porphyrin derivative is attached to a polymer or another nanoparticle to give a different applications. So to conclude, to summarize the talk, when we started this discussion, we called the porphyrin the pigment of life. So another thing is a ring of life. It's a ring structure. We found that it's a ring structure. So it's a ring of life. So starting from, say, the hemoglobin, which is in our red blood cell, which is having crucial role in the oxygen transport and the oxygen storage, and after that the different synthetic modifications has been possible in the case of porphyrin structure. A lot of researchers are involved in the synthetic modifications of porphyrin structures, and the synthetic modifications are making this porphyrin capable to give different properties which can be used for different applications, like as I mentioned, in biomedicine, in solar cell, and in adsorption properties. So it's ranging from it is having influence in our different aspects of our life, and that's why we can call it as a pigment of life and ring of life. And I hope this lecture will help the students to read more about. As I mentioned, I have given just an outline of porphyrin, and I hope this will motivate the students to read more about porphyrin structure and will attract the students to do research in porphyrin chemistry. Thank you.